Hello, everyone. I'm Dr. Rucha Sarvade. I am a senior researcher in SKB Lab, which is in School of Biological Sciences, Monash. I'll interview a previous and present honor student of the lab, as well as Dr. Suresh Kumar himself. And we'll walk you through what happens in the lab and what research is going on in the lab. So let's get started. Hi, Craig.
Hey, Richa.
You did your honors in SKP Lab. How was your experience?
It was a really positive experience. Well, it must have been because I stayed on to do a PhD for an extra 4 years afterwards. Suresh is a really good supervisor. He gives you a lot of room to grow and pushes you to kind of ask critical questions and to think scientifically. And he cares a lot about seeing you improve as a scientist over the time that you're in the lab. The lab itself was a really nice place to do my honors. There was a lot of people doing different projects that I could interact with, and I could always have a conversation or ask for help or go grab a cup of coffee with. So I'd highly recommend it.
Hi, Jordan. How are you?
I'm good.
So you are a honors student in SKB Lab currently. Tell us about your experience so far in this lab and the work that you're doing in this lab.
I started off just helping people in the lab, and I got to build relationships with them, and everyone was just so helpful and wanted to help me learn. And so I learned things like learning how to do molecular work and taking care of the model organism we use, Arabidopsis. And then I moved into Gen 390, which is an undergraduate unit, where I did a project of making mutants with CRISPR for SR proteins, which I'm doing projects involved with alternative splicing and regulation with that. And now I'm doing an honors project, which has taken a hit with COVID-19 pandemic. So my original project was to characterize mutants I'd made in Gen 390, which is a lot of molecular work. But now with Suresh's help, we've transitioned to a new project, which is computationally based. So I've been performing GWASs and trying to find links between alleles and changes in splice site usage. So still with the same project, but a very smooth transition with Suresh's help. It's been really good.
Thank you so much, Jordan. Hello, Suresh.
Hi.
Would you like to tell us what does your lab work on?
Okay, I'm going to take you to a virtual tour of the SKB lab, and maybe I can share the screen first for you so you can sort of see what we do. Okay, I want to welcome you to the SKB lab where science is fun. Okay, I had a group called Phenotypes to Genes and Mechanisms, and we are interested in basically 3 areas of research. The first one concerns about how plants sense and respond to changes in temperature. And we are one of the leading groups in this area. And in recent times, our work has taken us in the direction of epigenetic regulation that's associated with temperature responses. And in the second part, which is also somewhat related to epigenetic regulation, is associated with some sort of a translational work where we go from plants to human genetic diseases. So we have discovered in collaboration with Sridevi Suresh Kumar's lab in the School of Biological Sciences, trinucleotide repeat expansion in plants, which, last year we had a paper in Cell where we showed that the underlying mechanism of how these repeat expansions actually lead to downregulation of gene expression. And now we want to try and test the same ideas and principles in the case of a disease called Friedreich's ataxia. And so this work is actually done in the human cell lines with commercially available cell lines and so on. The last part of the work is actually associated with the splicing code. How do the plants or how do the organisms basically decide when and where to splice and form different, how alternative splicing operates. So we try to understand that using a new technique that my lab has developed, which we call a splicer, which allows you to quantify the splice site strength. For instance, if you look at this particular splice site, you have the next generation sequencing reads that map perfectly here, and there's a gap, and then it starts to map it perfectly again and here, which tells you that in these reads, this site obviously has used, whereas in these other reads, it doesn't. So you can sort of use this information and work out how efficiently a particular splice site is actually used. And then you can actually use this as a phenotype in a sort of an EQTL type of an analysis and actually get GWAS done. And this allows you to actually work out what regulates splice site variation. You can identify associated SNPs for variation in splice site strength. So this is something that my lab is quite excited about, and all the 3 areas of work involves genetics, molecular biology, and nowadays also some sort of a computational work. So that sort of gives you an overview of the 3 different areas of my research.
Thank you. That was a great virtual tour you have given us. Thank you so much.
Thank you. Thank you. Have a nice day, Richa. Bye.
Thank you.